Hello, my name is Andrey Prjibelski. I am from St. Petersburg State University, Russia, and the title of my poster is IsoQuant, isoform analysis and quantification with long error-prone transcriptomic reads. So, my poster is about IsoQuant, a new tool we developed for long-read RNA analysis. The pipeline is pretty simple. We take as input a gene annotation and long-read alignments. You can obtain those using your favorite spliced aligner, for example Minimap, Gmap, or Starlong. We further match mapped reads to known transcripts and try to assign each read to a known isoform, detect inconsistencies between reads and annotation, and count different genomic features. We further split reads into two different categories: consistent splice matches, which most likely represent known transcripts, and inconsistent matches, which represent novel transcripts or intron retention events. We group those reads by their inconsistency type and try to create a novel transcript model. Some of the algorithmic details you may find in the poster itself. Firstly, we tested our tool on simulated data. We used two different tools: isoSEC-SIM, which inserts errors uniformly, which is similar to what really errors look in PacBio data, and TransNanoSim, which learns on real data and inserts more nanopore-specific sequencing errors. For testing, we used reference transcripts as a perfect case. We simulated PacBio CCS data with error rate at about 1.5% and two different nanopore datasets, with isoSEC-SIM with uniform error rate and with TransNanoSim with ONT-specific errors, both 11% error rate. We tested IsoQuant against Scanty tool on all simulated data we generated. As you see on transcripts and PacBio data, both tools have nearly perfect precision and very high recall. However, when it comes to nanopore data, recall drops significantly and precision for Scanty also decreases. However, IsoQuant manages to keep high precision on both nanopore datasets. One thing I may notice is that mouse dataset was generated using TransNanoSim, so it's more realistic. And as you see, the difference in precision is more noticeable than on human data, which has uniform error distribution. The second experiment we carried out is assessing transcript model construction tools. Again, we simulated reads using the entire gene annotation and provided them to tools which are capable of constructing novel transcript models. However, we also provided incomplete gene annotation to these tools by randomly removing about 5% of spliced transcripts. Further, we compared the results of transcript model construction against the original complete annotation using GFF compare, which allows to compute sensitivity and precision. In this experiment, we compared IsoQuant against StringTie2 and FLAIR. Again, on PacBio data and nanopore data with uniform error distribution, you can see that precision and sensitivity have very high values. Probably IsoQuant have just a fraction higher precision than other tools. However, again, when it comes to TransNanoSim generated data, which is more realistic, you can see that both precision and sensitivity drop significantly. Positive thing for us that IsoQuant manages to keep very high precision and nanopore data at 99.4%. Of course, we also tested IsoQuant on real data. For this purpose, we used publicly available human PacBio CCS and nanopore direct RNA sequencing data. As expected, percentage of uniquely assigned reads in PacBio data is higher than in nanopores, and nanopore reads have more inconsistent reads. As expected, it affects the amount of novel transcript constructed. It is higher for nanopore data than for PacBio data. Of course, it is not possible to say which tool is correct and which tool is not on real data, since the ground truth is unknown. However, we try to estimate how consistent these tools are, and it appears that they are not very well consistent. For example, for PacBio CCS data, they have only 8.2% of transcript models generated by all three tools, and the number for direct RNA nanopore sequencing is even lower, 7.3%. What you may notice is that IsoQuant doesn't generate a lot of transcripts that are not supported by other tools. For example, for PacBioCCS, we have only 0.1% of transcripts which are not supported neither by StringTie nor by FLAIR. And the number for nanopore data is a bit higher, 3.7. Finally, as a conclusion, I may say that we developed a new tool which shows high precision for read assignment and transcript model constructions on both types of long-read RNA sequencing data. We still think that analyzing nanopore RNA data is a big and open challenge, and also we noticed that tools that generate transcript models based on long-read data are not very well consistent with each other. Thank you for your attention. I would like to thank my team, and if you are interested in our tool, check out the GitHub link. Thank you very much.